generation, then this makes this one the third generation of the Herschlag lab.
Yeah.
And our first speaker is...
I think this is called the degeneration.
Whatever it means. And our first speaker is Zalatan, University of Washington, and they told me, 15 minutes, not 50, 15, plus 5 minutes question, and at 13 minutes I will stand up and interrupt your talk.
Excellent, I'm ready for you. All right, thanks everybody. My title's a little bit different. There's a story there that involves last-minute control on the other project that didn't quite turn out the way we expected. So anyway, I decided to talk about something new. This is some brand new stuff in the last week or two. So it is also very raw. It's very possible that there's a logical hole right in the middle, and Dan's going to squish it up and down and say. But actually, I'm genuinely hoping that Dan stops. So one of my favorite phrases from Dan was, "I know what's going on." A good amount of the time, he was actually right. Anyway, all right. So I'm going to tell you a little bit about mechanistic questions in cell signaling networks. All right. Before I start, the two grad students in my lab who did all the work are Aaron and Mary. Give them a shout out at the end, but they did all of the experiments. Okay, so a big question that my lab has been interested in for a while is trying to understand how cells specifically process signals. And some of you may hear echoes of my graduate work. I was hung up on specificity at the level of the 8K superfamily when I was a grad student. As a postdoc, I got interested in thinking about sort of broader questions in cell signaling networks. And so the big challenge we think about in cell signaling networks is, so we know there are all these signaling molecules and inputs that activate networks of proteins inside the cell and lead to some sort of output. And the big question we have is, how do these signaling proteins know who they're supposed to talk to and find the right target? And this is a problem for a couple reasons. First, because a lot of these signaling proteins are homologous. There's places where signaling could crosstalk to other similar kinases and other signaling molecules. But a bigger problem has been caused by the genetics and genomics and cell biology revolution. Really, the fact that we now know that all of these networks are really, really highly interconnected. There are lots of places where proteins are shared between signaling pathways. You'll see talks where there's a blizzard of arrows talking to all the other arrows. So lots of things are used and reused in multiple contexts. So how are they specified for particular functions? One way we think cells solve this problem is they use these proteins that we've started to call scaffold proteins. These are molecular assembly platforms that have binding sites for multiple proteins in a signaling pathway. And the idea is they co-localize a set of proteins in a signaling pathway, bring them together, accelerate the reaction between these, and say preferentially promote reactions on one particular path in a signaling network. And so a lot of this data comes from simply doing things like co-IPs and identifying proteins that have these binding functions. There's actually very, very little mechanistic data that tells us what's really happening. What's actually, at the molecular level, what are these proteins doing to specify these reactions and control reactivity in these networks? So really what we know from a lot of good data, cell biology and genetics, is that there are these proteins that assemble multi-protein complexes, and then we infer a lot of functions from there. So you'll see this in a lot of papers. So they'll say, oh, it must be bringing things together, and proximity effects are accelerating reaction. Maybe as a consequence of this, you're effectively insulating these signaling proteins from other pathways, or perhaps by binding this particular scaffold, it prevents this protein from talking to things that are not on the scaffold. There's also thinking that these scaffold proteins are localized to specific places in the cell, so they drive localized signaling. But again, really what we know is that bad things happen when you mutate these things. You knock it out, pathway is dead. So we're really missing mechanistic molecular detail for what exactly is going on in this cartoon picture that is, what exactly is happening here. So my lab has been attacking this problem, and particularly we're interested in this notion that proximity effects accelerate reactions. What does it actually mean to say you bring two proteins together, the reaction gets faster? How much faster? How does that depend on the structure? And is this potentially a point of regulation? If it's precisely dependent on structure, can you then imagine regulating scaffold protein structure in a way to tune reactivity in the signaling networks, both from an engineering perspective and from a biological regulation perspective? So my lab is working on this project in sort of two areas. The original title referred to this idea we have of trying to engineer scaffold proteins from the ground up, a minimal model system. You guys have heard that one before. That can be used to, basically map out what is the relationship between structure and function by building what we think are representative molecules and trying to tune the distance, the flexibility, the orientation of these molecules and see what happens in reactivity. Weird things are happening in this project right now. So we're going to talk about this project this time.
They don't sound so much more interesting.
Yeah. I'll have a whole other talk to you later. So we've also been trying to study natural scaffolds, and this is interesting because these are molecules that are clearly important biologically. They're also hard to study. They're complicated, big molecules, many different domains. They're hard to express. They do a lot of weird stuff. They are post-translationally modified. They undergo conformational changes. So while this is really interesting, and we want to think about what these things are doing in a native context, there was a reason why we were trying this sort of minimal system. We like minimal systems. But anyway, we're done. Well, we're not done, but we're done for now. So the model system that we've chosen to focus on is the Wnt signaling network in mammalian cell development. This is a really important pathway. I'll just give you a quick overview. I'm stripping some of the parts, but the way this pathway works is that there's this protein we're calling a scaffold protein, axin, that binds this kinase GSK, and it phosphorylates this molecule beta-catenin. Beta-catenin, when it gets phosphorylated, gets degraded. So this is the off state of the pathway. It's kind of a bizarre off state because what it means is the cell is constantly making these proteins, constantly burning ATP to phosphorylate them, and then breaking them down. That's kind of energetically costly. We can speculate about why that's a good idea. When this pathway gets turned on, the signal ligand, a Wnt ligand, binds a receptor outside the cell, which communicates to this protein disheveled. There are a lot of stupid names in this pathway thanks to the Drosophila geneticists. So this has something to do with a disheveled-looking fly. I don't really know what a disheveled-looking fly is. Anyway, thanks Drosophila people. So disheveled communicates to this scaffold axin, and somehow through these things that are mechanisms that are again still sort of mysterious, that stops this phosphorylation reaction, beta-catenin accumulates, it can then go to the nucleus and set off transcription and activate the pathway. So we got interested in thinking about, oh, this looks like an interesting place where there's a scaffold that seems to be itself subject to regulation that might be tuning reactivity in a kinase network. Can we understand first how is axin contributing to the reaction here, and then what's changing to cause it to stop driving this reaction? There's a hint of data in the literature from a fairly prominent paper that suggests actually a conformational change could control what's happening here, but again, sort of short on details, but interesting and suggestive. And the idea is there's actually one domain in this big scaffold that is important for this reaction. It's normally phosphorylated. When the pathway gets activated, it gets dephosphorylated, and that drives this intramolecular reaction that, again, through actual molecular mechanisms that remain mysterious, something about this closed state is incompetent to do the phosphorylation reaction. So we want to, again, sort of start picking this apart. Can we make these molecules in vitro, measure reaction rates, and start manipulating with mutations and truncations and try to map out what's actually happening in the system? Okay, so the model that we're trying to think about is that this axin scaffold accelerates reaction by tethering these two proteins together. There's some old data for this. So about, I guess, 15 years ago, some people purified these proteins in vitro, added just a kinase and beta-catenin, measured reaction rate, and then added axin and measured a reaction rate and said the reaction gets 24,000-fold faster. Wow, that's big. That's an interesting place to start to get a foothold. But we said, wait a minute, we should probably read this paper and just make sure we know where this number came from. Okay, so they report single time point kinetics, so there's no time zero, there's just product, okay, and that makes a rate, 1 point makes a line. They pick, arbitrary concentrations that kind of don't make sense, but anyway, so there's weird stuff going on here, but anyway, they have an observation that there's a 40-fold increase in the observed rate. Then they say, okay, well, we think that only 1 in 3 of these scaffolds is actually binding beta-catenin, so we need to multiply this number by 3. Oh wait, only 1 in 200 are binding GSK, so there's a vanishingly small quantity of the assembled scaffold in this reaction, and that gets us about 24,000. I could spend a really long time going into detail, but this is sort of my executive summary of what's going on. There's just so much messed up about this. So anyway, this one really gets me. I cannot understand where this number came from. I've been bugging my students to explain it.
Have you presented this in any outside?
No. I made this picture for my own entertainment, and I thought you guys didn't see it.
So outside it may not be funny.
Yeah. we are back way at step 0 now. We thought we sort of knew what's going on. We need to actually measure rate constants. And really, the big thing that these guys screwed up is they never actually defined the thermodynamic and kinetic framework. They never actually measured rate constants. We need to make a well-defined comparison here. This actually gets complicated really quick. So we've got the scaffold protein, we've got a kinase and a substrate. Either one could bind, and then it can assemble to this ternary complex that reacts, and then it falls off. All right, this is not terrible. Some of you RNA people showed me way scarier schemes back in the day. Things get a little bit irritating here. You have to be careful with your protein concentrations. If you go to too high scaffold concentration, we think what might happen is you have so much excess scaffold that, say, each scaffold is only binding one of the partners, and if they need to co-assemble, then you're going to essentially stop the reaction by doing something like this. So we need to simplify this scheme. And one thing we know from the literature, and we've corroborated some of these numbers, is that the binding constant for the kinase is much, much tighter than the binding constant for beta-catenin. And what that lets us do is it lets us, at least for the first couple assays, do our experiments under conditions where we think we have enough axin to bind GSK, but not enough to bind beta-catenin. So we have this simplified scheme where we're starting with this pre-assembled complex. We're going to measure steady-state rate constants and hopefully get out parameters like kcat over km and kcat, and use this and then compare to a minimal reaction without the scaffold. So we can, again, do the same sort of experiment, put everything together, measure rates, and compare kcat over km values, or kcat values. By doing this, we prefer kcat over km. Some of you may remember this, again, back from my graduate school days. Kcat over km is way better than kcat for so many reasons. One of the more important ones is it's a well-defined ground state. We know what we're looking at. And so in this case, even though intuitively you might think if we want to look at a scaffold, we want to look at the ternary complex and go from there, what this number should tell us is something about how this extra binding interaction contributes to transition state stabilization. Okay, there's my alpha get energy diagram. Okay, so we're going to do kinetics, and now Mary and Aaron will be mad at me because I just skipped over like months and months of work of them figuring out how to purify the proteins. This is hard. There's a reason why these seem like really basic questions, but nobody has done them even though we've understood these pathways for a while, or we think we know about these pathways for a long time. So purifying these proteins is tricky, especially when your freezer breaks. And anyway. So we can measure rate constants, and it turns out when we measure rate constants, so what you're looking at is the observed rate. We're increasing the substrate concentration. The rate actually increases linearly, so we don't actually see saturation. We can measure kcat over km. It's actually a reasonably big number. But what we want to see now, okay, what happens when we measure the rate of the scaffold? So again, we're picking a single scaffold concentration that's between these two binding constants, so we think we're starting in this state. And when we measure those reactions, we actually see significantly faster reactions. So this is cool. The reaction gets faster. At very high substrate concentration, it levels off. The fit is not great, and honestly, we're a little bit suspicious about whether this is really saturation or whether our proteins are starting to get mad at us. So we don't want to get too hung up on kcat here for a lot of reasons. We can look at kcat over km, and we can see that there's a 100-fold increase in kcat over km. So this is cool. We see a well-defined increase, or increase in a well-defined rate constant. This is a big number. It's not 24,000, but it's still probably good enough for biology. We do a couple other important tests. So we want to vary the axin concentration. We have an expectation that starting from low axin concentration, the rate should be slow, and then it should get faster as we're adding the scaffold in. But at a certain point, when we cross the concentration of GSK, and as we start going higher and higher, the reaction should slow down, and we do see that. So that's gratifying. Okay, but then we do one more key control experiment that the little Dan voice in my head told me was really important to do. Oh wait, I'm sorry, I forgot to say that. So this is a picture that Dan has the top. This is what we think is happening, right? So at this low concentration, we've got one bound, but at too high axin concentration, they're separated. Okay, so now little Dan comes along and says, you need to do one last important control, and that is to vary the enzyme concentration, make sure you're actually measuring a biomolecular rate constant. So what you're looking at here is data for this plus axin scaffold reaction. We're using kind of a high concentration, but you see it looks more or less linear as we increase the enzyme concentration. But now things start to get weird because we do the exact same experiment for the non-scaffolded reaction, and it's not linear. And it's not linear in a weird way. It looks like down at these very, very low enzyme concentrations, it's actually pretty similar to the plus scaffold reaction. So at the concentrations that we often work at, is that my 13 minute? Or almost there. At the concentrations we're normally working at, what we may actually be seeing is some sort of artifact, something weird is happening at high enzyme concentration. The simplest model you could come up with is maybe actin is active in the monomer state, but at high concentrations, it's either dimerizing or oligomerizing, and we actually do have gel filtration data that backs that up. So this is kind of troubling, and this is where we were last week. So what's actually happening is maybe GSK is switching between this active and inactive state, axin may be driving it or stabilizing this monomeric state, and now we actually need to go back and redo all that kinetics we did, because now I no longer believe that 100-fold number. We need to go back and pick the concentrations for them carefully. My guess is, based on what we've seen, it's probably closer to 10-fold. It's definitely not 24,000-fold. But again, so we're still back at square one. What is this scaffold doing? And then we can start trying to figure out how it's regulating. There may or may not be something interesting here about this inactive state. It may be an artifact of our in vitro conditions, or it might be an interesting regulatory mechanism that we hadn't really thought about before for scaffold proteins to stabilize an active state of the target. And then moving forward, we have some other questions. So I didn't even talk about this mystery molecule. APC, it stands for Adenomatous Polyposis coli. It has a weird name from cancer genetics, but this is the molecule that gets mutated all the time when you get colon cancer. This is a nasty thing, or well, it's nasty when it's mutated. Anyway, we're interested in understanding whether this molecule is actually contributing to making the reaction faster. And so we can start purifying these things and assembling more complex models for in vitro reactions and seeing what's happening once we've got this well-defined system going. So we're at the early stages of this project. We think we've got an interesting handle on the scaffold that might teach us about how these kinase networks are regulated. And with that, I will thank Mary and Aaron who did the work, the rest of my lab. Nice people gave us money. I do need to thank Dan. Big thank you to Dan. This is my favorite Dan picture. I showed you my thesis defense in this room. I did bring my lab notebook. Some of you guys know what that means. We'll talk about it tonight at dinner. Big thank you to Dan. In all seriousness, I think the biggest thing that Dan did for me was he helped me to learn how to think rigorously. He pushed me really hard in a good way, and it pushed me to think about how to think about scientific questions and how to structure rigorous logical arguments. This talk notwithstanding. I do also want to just give a quick thanks to the legendary alums, particularly Gita, Pat, and Shuo, who at various points in grad school and postdoc and beyond have given me really useful scientific feedback and advice. I've talked to various others. Katrine was wonderful when I went to a conference this summer, so it's been really wonderful to take advantage of the network of alums that I didn't actually ever overlap with, but have still been incredibly helpful. And you can see the echoes of Dan in their backs too. It's great. So anyway, thank you, and I'll take your questions.
Questions?
Jesse, great talk, and what concentration do you think GSK is at in the cell, and how does that relate to sort of the KD that you can estimate?
I'm not aware of a good measurement of that, I would say, and so my intuition honestly comes from thinking about these questions in yeast, where a lot of these simulating molecules are typically present in the mid to low nanomolar range, but again, that's just speculation from thinking about kinase networks in yeast. I don't know what it is in a mammalian cell.
And your sort of approximate KD is
so the
tens of micromolar or something?
So there's a couple relevant KDs here, right? So this one is pretty reasonable. This is actually comparable to the scaffold system we used to look at in yeast. Most of the binding constants are in the low 100 nanomolar range, and things are often present at concentrations similar to those binding constants. This is a bit surprisingly weak, which is part of the reason why we want to look at other components of that destruction complex. It's very likely that that other molecule that I didn't draw here is actually acting sort of as a co-scaffold to bring that in and maybe tighten this complex up.
So what was the concentration which you had?
Down in the diameter. Right, this is 100 nanomolar right here.
So you should be able to maybe get a really, really good estimate of the concentration just by the RNA levels that which are measured a million times.
You just said something super controversial.
Well, but you want an order of magnitude.
We can talk about that.
I guess maybe that's less important than the fact that this dimer versus the scaffold KDs are within twofold of each other, right?
Yeah, no, that's true.
So
it's happening on the same order of concentration. You think it should be binding to the scaffold.
So would it be possible to design fusion proteins to further simplify the framework?
Tricky. people have tried to yes, there are ways to think about doing that, and that comes up in some of the engineering schemes we've had to build some of these things from scratch. Working with these natural systems, it's hard enough to express the scaffold protein. Tagging things on the end is a little tricky. People have tried to do things like insert leucine zippers and then using synthetic recruitment domains to try to see if you can recapitulate the same effects with orthogonal interactions. But so far we haven't done that here.
One question and one comment.
Comments.
Only one. So the slide you showed where you have where you did the very nice experiment that of the rate going up and then down as you added more. Can you extrapolate that out or extend that out to see whether when they're bound to separate components, whether they react with the same rate that they do when they're free in solution? Because it might be that even just being in the complex is inhibitory in some way.
Yeah, so we have not quantitatively tried to model this. We really should. Actually, I have a copy of Kintech in the lab that I've been trying to convince my students to learn how to use. Honestly, the thing that worries me more about this picture is that if this binding constant for beta-catenin is around 1 micromole, things are falling off a little bit faster than I would intuitively think they should, which makes me worried that maybe, like, yet another complication is that a very high concentration axon may also be a ligand resin, so this is going to get complicated really fast.
So the comment is on the slide, which was one of your introductory slides where you showed the scaffold, and then, that's fine. Okay, so you showed the scaffold as this orange box.
Yeah.
Then in a later slide, you showed it as 3 separate pieces, right? And it's remarkable how much pictures affect how we think about things. And I'm not blaming you at all, but this is a field, right? How bad the pictures are that we usually use in these models. I think that's part of your point. And so I want to urge people to look up an artist whose name I've forgotten. But it'll be easy to find them. They take cover page 1 of the New York Times and change the size of pictures on it and the placement of things. And how you think about what the news was totally changes. And so I did this, for example, for the Charlottesville headline, news of the Charlottesville
It's getting dark really fast. Yeah. It's getting dark really fast.
So I think anyway, so that's a great depiction of how much we're affected and don't realize how we're affected by pictures. And I think it's something to consider more in science, how sometimes these pictures are limiting us, especially the pictures that are typically used for
Actually, I will say, as a more general point, I have a whole separate older talk where I talk about how bad these pictures are because they make you think that these are stable complexes. And even 100 nanomolar complexes coming off on a really fast time scale. It's particularly problematic in this field where people make arguments about binding the scaffold and then insulates them from talking to other things in the cell. If they're coming on and off on the time scale of cycles, it doesn't.
Okay. There are no more questions or comments. And we will have then